This is everything you need to know about the electron transport chain for the MCAT. And if you like this content, check out the links in my bio to the self-paced course or the MCAT Mastery Program. Both of those give you access to over 500 pages of the highest yield MCAT content just like this. We are going to start by looking at complex 1 and complex 2. First, let's establish that this is the inner mitochondrial membrane. That makes this the inner membrane space, and down here is the matrix. We're going to start with looking at complex 1 and complex 2. At complex 1, you have NADH arrive, and this is mostly being made in the Krebs cycle, and it's going to transfer 2 electrons to this molecule of Q. Q is called ubiquinone. Once those 2 electrons arrive at ubiquinone, it's going to become a molecule of QH2, and that is called ubiquinol. During the transfer of those electrons, that energy that is given off is going to be used to pump 4 hydrogens into the inner membrane space. The very same thing is happening at complex 2 with that transfer of electrons, where it turns a molecule of Q into a molecule of QH2. But this time, the electrons are coming from FADH, and there are no protons pumped at this spot. Now let's look at what happens at complex 3. We're looking at 2 steps here, so we're going to zoom in. First, we have 1 molecule of ubiquinol arrive, and it's going to transfer 2 electrons, but they're going to go to separate places. The first electron is going to go to this molecule of cytochrome C, and it fully reduces it into a molecule of reduced cytochrome C. Simultaneously, what is waiting at complex 3 already is this other molecule of ubiquinone. One of the electrons from the molecule of ubiquinol is going to go to that molecule of ubiquinone that's waiting at complex 3. And the ubiquinol that donates its 2 electrons then turns into ubiquinone. And then the ubiquinone that was already here gains 1 single electron, and then it just waits for step 2. And during this step, 2 hydrogens are pumped. For step 2, we have another ubiquinol arrive. It's going to do the very same thing. It's going to donate 1 electron to cytochrome C, which creates a fully reduced cytochrome C, so now we have 2 of those. And the other electron goes to that ubiquinone that is still sitting there. So now that ubiquinone has gained 2 electrons, and that turns it into ubiquinol. So notice now it is QH2. The ubiquinol that arrived turns back into ubiquinone. So during 2 steps, we had 2 ubiquinols arrived, but we replaced 1 because we created a ubiquinol right there. So we had a net change of just 1. And then we also took 2 cytochrome Cs and created 2 reduced cytochrome Cs. And during that, we also pumped 2 hydrogens per step. And now we can look at complex 4. Complex 4, let's zoom in. We have those molecules of cytochrome C that arrive. Complex 3 only produced 2 of these with those 2 steps, and we are going to require 4 cytochrome Cs to fully do this step at complex 4. Each cytochrome C donates 1 electron. So after 4 electrons are donated, they are going to combine with a molecule of O2 and produce 2 water molecules. And during this process, 4 hydrogens are pumped through complex 4.